Hello everyone, my name is Claudio Fernandez. Today I'm here to talk about my research for my PhD that is based on disease detection in potatoes and also in cucumber greenhouses. So today specifically I will talk about applying remote sensing to potato lake blight detection. So today I'm here to talk about late blight. This disease is caused by an oomycete. The Latin name is Phytophthora infestans and is the pathogen that caused the Irish famine, causing around 1 million deaths. This is important for Canadian economy because in the Atlantic, the provinces of New Brunswick and Prince Edward Island are producing around the 40% of the total surface of potatoes. Also, the disease has a huge economical impact to avoid infection in the fields. Why using remote sensing for disease detection? Because at field level, the applications are accurate and time and cost effective. Also today we have drones that we can uh, connect to multispectral or hyperspectral cameras, and we can have maps of the fields where we can identify different areas with different kind of stresses. Specifically, diseases at early stages causes physiological disturbances that can be detected by remote sensing, and these are produced in the visible region of the spectrum, specifically because changes on the chlorophyll content, and also in the near infrared region of the spectrum because the disease will induce structural changes on the leaves. Here we have the typical leaf reflectance of a healthy leaf. We can see there is an increment from around 660 to 780 nanometers. This region is the red edge region and is an important region of the spectrum because it's really sensitive to stress and can be used for disease detection. During this video, I will summarize the research conducted in a walking chamber in a biodrome located at University of Western Ontario. Also, I will show you the changes in the reflectance at the leaf level induced by the disease from the moment of the inoculation, the potential wavelengths to detect the disease, the relation of this hyperspectral information to a commercial sensor, the best vegetation indices to detect the disease, and the models that we determine for the red well point and the red edge point. On the right hand, you can see the MicaSense red edge camera. That is the commercial sensor that we'll be using as a reference for future field work in the Atlantic. The methodology consisted in growing plants. Those plants were inoculated using Phytophthora infestan solution with sporangias, and then the plants were placed in the conditions necessary to induce the infestation of the leaves. Also, the hyperspectral data was collected with an ASD spectrometer, as the one that you can see here in the image, and a contact probe with a leaf clip were employed to collect data at the leaf level. This is the workflow that you need to use to compute the red well point and the red edge point. As you can see, it's very extensive. That is why I am not showing the formulas that I use to compute all the parameters that I will be evaluating, but I will suggest you to have a look at the papers that I will tell you at the end of the presentation, so you can have a look on them, have access to the formulas, and also have access to the computation of the different vegetation indices. The results related to the red well point shown that this parameter adapts better to a linear model. On the other hand, the red edge point showed that exponential model fits better to the data. Also, we detected that one day after the inoculation, the value of the red edge point of the infected and the healthy leaves presented significant differences. Here we can observe how the mean reflectance of healthy and inoculated leaves changes since the moment of the inoculation. 3 days post inoculation, that was the time where the disease was detected for the first time. And we see there that there are some changes around 550 nanometers and a little bit around 680 nanometers. And at the end of the evaluation, we can see there is some major changes between 400 and 500 nanometers, from 550 to 680 in the reddish region and also in the near region. Because the previous figures were representing 501 variables or wavelengths, then we applied a principal component analysis, and then we determined that the first three principal components were conserving most of the variance of the data. Also, here we can see again that 3 days post inoculation, the data start to group, like to mark trends between the healthy and the infected leaves. Also, we can see that the first component shows the highest values, and these start to decrease according to the development of the disease. At the same time, the second principal component increased its value from 3 days after inoculation. And we also detected some increment in the third principal component, but actually I used it just because I needed to improve the quality of the plots, plotting 3D plots instead 2D plots. In order to detect potential wavelengths to detect the disease, we compute the spectral ratio with the hyperspectral data. The problem with this technique is that on each day, the peaks on each regions are changing according to the disease development. Therefore, it wouldn't be suitable to collect information from this analysis to have a unique wavelength for disease detection. That is why we simulate the mica sense bands. This is the location of the 5 bands of the mica sense reddish camera. We can see some information regarding the range of each band, the bandwidth, and you can visually see where it's located in the visible spectrum. So considering that we simulated the reflectance of the bands of the commercial sensor, we can see immediately that the near band is not good to detect the disease. Basically because of the change in the slope at 840 nanometers in the spectrum. And we can see that the other 4 bands seems good for the disease detection. 3 days after the inoculation, when we see the symptoms for the first time, we have a 5% of difference between the day of inoculation. So that means that we should have a 5% difference in the reflectance between an infected and a healthy leaf when the disease is visible for the first time or in the early stages of the disease. Now here we can observe the ratio variation of different vegetation indices. And vegetation indices are computed because they allow to show better some differences in the properties of the plants than just the raw reflectance. We can see that in the group number 3 and the group number 4 we have the better indices because these are the indices that are showing a higher variation regarding the day of inoculation. For example, the redness index in the group 3 days after inoculation has almost a 20% of difference respect the day 0. We provide the first study using hyperspectral data to characterize the disease. Also, we provide the first study modeling the variation of the red well point and the reddish point. Also, we provide the detection of the best bands in relation to the mica sense camera and the vegetation indices that could be used for late blight detection. Also, the percentual differences with respect to the healthy leaves, that can be used like a threshold to create a discrimination between healthy and infected plants. How do you do that? Basically, in the field work, you collect images with the drone, you remove the soil effect, and then you can create a pixel by pixel analysis. And if you detect differences over 5 or 10%, depending if you're using the raw bands or if you're using the vegetation indices, you can deduce that you are having problems related to late blight. These are the suggested readings that I mentioned earlier. So it could be interesting if you can have a look on them because you can see more figures, you can see all the formulas, you can see the full methodology, and also you can see the difference between the leaf results that I showed you here against the canopy level results that is using the whole plant, not only leaves. Also, you can have a look on the machine learning algorithms to sort and classify the data into healthy and infected spectrum. So it's a more complete and you can have a better idea of different possibilities of that analysis to detect the disease. Finally, I would like to acknowledge to all the team that has been working during this project, especially my advisory committee and also the technical help that I got from people from A&L laboratories, specifically to both Chan because he helped me to set everything inside the Biotron and to get some materials and move them around the city, and to Julia Körner because she taught me how to grow the Phytophthora infestans inside the lab and to keep them alive in vitro to finally get the inoculum to be able to study my project. Finally to Dr. Benoit Bisimungo because he provided the first tubers to start these experiments in the Biotron. So I hope you enjoyed this presentation. Feel free to contact me and also to have a look on the papers. At this moment on my PhD, I'm changing the project now to cucumber diseases with multispectral images. So maybe next year I could tell you about that. Thank you.